Okay, students. So very good morning. So after the last class of where we have discussed something about disaccharides, now it is the time to move up to polysaccharides, right? So actually, we all know that the word mono comes from single, di comes from two, so oligo comes from few, and poly means many. So here the time is to study about polysaccharides. Poly means many, saccharides means sugar molecule. Alright, so if you see, sorry, before I move on to the details of this particular molecule, just see the structural arrangement over here. Again, students, see the structure of glucose. It is all association of glucose together. Give it the time, look at it. All is 1 4 1 4 1 4 1 4, right? This is just an example. I mean, what it is, what it is not, I will let you know, but mainly plants and animals, these two things are considered to study about polysaccharides. So you know that plant has the ability to synthesize starch via photosynthesis. So starch is generally representing the carbohydrate in case of plants. You have seen rice, you have seen potato. There are so many starch accumulation in all those products. So if I talk about starch, so most of the part of a starch is built up of amylose and amylopectin, right? So this association of amylose and amylopectin together is giving rise to starch. And what is amylose and amylopectin is, it's nothing, it is association of glucose, but there is a difference between amylose and amylopectin. Amylose is a straight chain of glucose molecules, glucose polymers with alpha 1,4 linkages. Same thing that I have discussed earlier also. This is amylose, which I have shown you just now. So amylose is what? Association of glucose molecules in alpha 1,4 linkages. Now, in what is the specificity of amylose? The end of this particular polysaccharide, where this anomeric carbon is not involving with any next association, we call it as reducing end. Now this particular C1 is not available to make bond with any other incoming molecule. We call it as reducing end of it. Fine? Okay. So jahan par bhi this last molecule is having C1, which is free, it is a reducing sugar. Okay, fine. So let's move on to the next section. Yeah. So after amylose, amylopectin. Now see students, this is amylose, this chain is amylose, and you have amylopectin. What is amylopectin? It is found at the branches, branching. Look at this association, alpha 1,6. Look at this, look at this. So the carbon belonging at C1 position is associating itself at the branching position. Now look at this, that is what I want to show. I hope you will be understanding what I want to show. This is what the branch is. I have depicted in red color the position number 6. Now this is busy with bonding to the upper sugar molecule. This is this one. At which position? At 1. We call it as alpha 1,6 position. Fine. So the difference between amylose and amylopectin, that yes, the sugar molecules are same, the bonding is glycosidic bond, but the configuration is, in amylose it is alpha 1,4, in amylopectin it is alpha 1,6. So don't forget, this is the specific difference, and make sure more is the branching, more is the complexity. However, you should know that the branching is somewhat also larger as compared to smaller, I mean these straight chains. Okay. So why branching? Because it produces a compact structure and it provides multiple chain ends so that enzymatic cleavage can occur. I will discuss about this in my Zoom session also. Okay. So I have discussed about amylose and amylopectin so far. Fine. So, the next molecule after starch is glycogen. Glycogen is considered to be the human starch, I mean your energy reserve. Glycogen is a glucose polymer storage form of energy in animals, in us also, right. But the difference is that it has more number of alpha 1,6 branches, more number of alpha 1,6 branches. So, you can understand the significance of branching, right. So, when more of branch is present, there will be more complexity. So, which is going to be more complex? Glycogen is going to be more complex. So, what happens when this kind of a structure is prevailing? All this highly branched chain structure permits the rapid glucose release. So, that what happened is that you cannot break easily the glucose from a straight chain, but at branching they are quite free, you can break them out. So, you need energy at an instant when you do some physical exercises, etc. So, it is easy to obtain glucose from this branching. That is why nature has chosen glycogen as your energy reserve also. Okay. So, next move on to cellulose. So, after glycogen, cellulose. Those who have studied plants or any agriculture student, so you know very well about cellulose. Cellulose is a constituent of cell wall to provide the strength and rigidity. It consists of long linear chain of glucose, but the difference is, yes, it is beta 1,4, right. So, you should be, you are now in a better position. Yesterday I have discussed about the arrangement of alpha 1,4 and beta 1,4. You can see all bonding over here, that is in flip flop arrangement. It is beta 1,4 linkages. Yeah. So, because of this beta linkage, there is a there is a strong promotion of inter and intra chain hydrogen bonds, van der Waals interactions, which causes the cellulose chains to become rigid and straight and packed with a crystalline arrangement like this. This is called as microfibrils when the cellulose are arranged like this due to flip flop arrangement of bonding, right. And because of this, the plant cell wall is quite tough, fine. All right. This I have already discussed. It is, the role is to impart strength and rigidity, which can withstand high hydrostatic pressure, right. So, it prevents the osmotic swelling. Okay. Fine. Now, next one is somewhat complex. So, I will be discussing in my next uploading material about this. Thank you.